multi-phase flow in cross media. So Yves, the floor is yours.
Thank you. Thank you. So it's really a pleasure to be back here. Thanks to the organizers. So I'm going to tell you about how flow-induced stress impacts phenotypic heterogeneity in bacterial growth and adhesion on surfaces. So what we mean by phenotypic is, for bacteria, is simply is related to their behavior independently of their genetics. So it can be impacted by the genetics, but not necessarily. And in that case, the bacteria will be all isogenetics. They're all exactly the same, but they do not behave exactly the same. Okay, so we're gonna basically analyze images of bacteria growing on surfaces like this. So they're biological objects. They are visualized with microfluidic experiments, and we use a physics-based approach to infer information about the biology, but we don't do biology. And so, of course, the motivation is hydrogeology related. Okay. So the environment context of this work is as many things we do in our team in Rennes, is that be it the fate of elements cycling through the shallow subsurface, or be it something that relates to the clogging of geothermal fields it's not biofield, but it's geothermal fields. It's what happens at small scales can play a role in controlling large-scale processes, and bacteria are ubiquitous in these things. So what we do in Rennes is usually we try to do experiments. We've worked a lot with the 2D systems. So examining multi-phase flow, velocity field, solute transport, mixing, reactive transport, and so on. I've put on purpose images from old work from 10 years ago, and you'll recognize some names. So that means that in 10 years, hopefully some of you will be speaking here and will be organizing the conference. So now if you consider that bacteria in a porous medium consuming some nutrients that are being brought, advected, transported by the flow, you can consider this as reactive transport, right? The consumption is a reaction. And so you're going to have a coupling between flow, transport, and the growth of the bacteria, of the population. So this was basically the objective of Antoine Hubert's PhD. And so we could start examining those couplings numerically, but experimentally we realized really quickly that when we work with bacteria, it's soon very, very complicated. So in fact, what we're going to be working is simply the growth of bacteria on a surface in a straight channel. And luckily, we found interesting things to report on. So bacteria have a very complex behavior in the flow and near surfaces. So they can be motile or non-motile. That means they can move by themselves or not. They can attach to surface. They can move on the surface. They can attach firmly and divide and grow and grow biofilms. Those biofilms will then make streamers and so on. You've seen some talks mentioning those aspects. And all this over time scales that vary from 10 to the minus 1 seconds to 10 to the power 6. Okay, so we study. We are in this range, so from up to 5-6 hours, basically. And what we investigate is simply particle division and attachment to surfaces on the flow, considering times at which only one layer of deposited bacteria is present. So we are far before the creation of a biofilm. So it's an extremely simple, the principle is very simple. So the bacteria that we study are the E. coli, so possibly the most used bacteria for experiments. So they are tubular like this. They're 2 micrometers long prior to division, 1 micrometer wide, They are found in the guts of warm-blooded organisms. They can be harmless or they can be dangerous. They can be, so they are aerobic, so they breathe oxygen, but they can also survive without oxygen. They can be mobile or they can be not mobile, depending on the strains. And so in our case, they're motile, sorry. So in our case, they're non-motile and they're harmless, of course. So they're prepared in a very standard way, in a very reproducible way, of course. So maybe I don't need to give you the details of. And something that's going to be important here is that we can visualize and measure the division of individual bacteria. And when we do so, we basically will fit an ellipsis to this bacterium, and we're going to get the center of gravity, and we can also get the 2 poles of the ellipsis. And so we can follow the displacement of those poles and of the center of gravity. And what we see is when they divide, the pole that was inherited from the mother bacterium, so this bacterium will divide, will grow, and it will divide in 2 bacteria. And in fact, one of the poles here is more firmly attached to the surface. And so it will, the growth will occur from the other side. Okay? So, okay, so you don't see the cell. Okay, okay. So we're using soft lithography cells, microfluidic cells. So they're made of 2 channels. So I'm going to draw it here. So you have basically one channel where the bacteria are going to be injected and we're going to be monitoring. And there is another channel on top where we flow oxygen. And they're divided by 150 micrometer thick layer. And so that allows oxygen to diffuse so that the bacteria are never lacking oxygen. They always have enough oxygen. And they also always have enough nutrients that is in the solution that is being flowed. Okay? So we use an inverted microscope with an incubator. We are at controlled temperature, 37 Celsius degrees, and controlled humidity. We first inject the bacteria into the cell. After 30 minutes, then we stop the flow for 30 minutes to let the bacteria attach. And then we re-inject a clean nutrient solution for 15 minutes at the desired flow rate. And after these 15 minutes, we start measuring. Okay, still with a steady injection. So we know what the consumption rate of those bacteria are. So they are in the literature. So we can compute a dam color, both for the consumption of O2 and the consumption of the nutrient in the solution, and they're always really small. Okay, so that means that we have no limitation to the dynamics of nutrient due to the consumption, to the dynamics of nutrient or O2 consumption. Okay, so it's not the transport of the nutrients or O2 that limits the consumption. All right. So we then analyze pictures. So we find basically individual connected clusters here that are then analyzed in terms of size to find bacteria. So here the dark blue are just one bacterium, and when it's light blue, it's 2 bacteria. So there are very few that actually contain several bacteria. So we're able to. So we can, in any case, count all of them in this manner. All right. So we make experiments at different flow rate. That means that we impose different shear rates. So the shear rate is simply the derivative of the velocity with respect to the vertical direction, because the confinement is mostly vertical. It's a channel that is thin, and much wider than it is thin. And the shear stress felt by the bacteria is basically the viscosity of the fluid times its shear stress, average over the first 3 millimeter over the surface, given the size of the bacteria. Okay? So we will work with 4 different flow rates and therefore 4 different shear stresses, which I will denote U low, low, med, and high. Okay? So these are the shear rates from 9 inverse seconds to 116 inverse seconds, and shear stress from 5 millipascal to 80. And so we will try to understand how what happens when we under the effect of those shear rates. So this is the initial state for the 4 conditions, right? And this is what happens in the U low regime, so very low shear stress. Okay, so we see the bacteria dividing, and their color here indicates the time interval of their birth. So the mean division time that we measure is 40 minutes, and so the dark blue are those who were born during the first time interval of 40 minutes, and the red are those who were born in the last 40 minutes. Okay, so we see that most of the bacteria will eventually they will divide several times, and in the end we mostly have red. Okay, and we have covered the surface homogeneously. I forgot to say that okay, so we have about 200 pixels per bacteria, so we can really quite well let's say resolve those bacteria, and we are acquiring at 1 image per minute. Right? So now we increase the shear stress. We are in the low regime, and we see that we're having a more sparse spatial distribution, and the temporal distribution of event is also larger. So we still have some dark blue that remain much longer, and you will see that at the very end, so we're getting to the end now, we are at 355 minutes. So we have basically every color. Now if we again increase more to this med regime, we will see that only a few colonies grow. Most colonies do not grow. Most bacteria do not divide. And we get these, we get actually elongated patterns. So the bacteria tend to grow in the back of the cluster, where they're kind of a bit protected from the flow. And we have much fewer freshly formed bacteria. So we start having a lot of blue and dark blue. And so when we go to the high regime, we have, let me see the time, we have an enormous amount of blue at the end. So the old bacteria dominate.
And did you check that none of them got filtered out? They weren't making...
Wait.
Oh.
So, of course we did. So, okay, so I have to hurry up a little bit. So this is the final state. So it's pretty obvious that the behavior is relatively different and varies monotonically with the shear stress. Okay, this is for the 4 different shear stress as a function of time. Okay, so clearly shear stress induces heterogeneity in the division rates. And so now we look at the total, the number of bacteria normalized by the initial number here in log scale for the 4, and we look at the same for the bacteria that were born in the different time intervals of 40 minutes. Okay, so you see that when we increase the shear rate, the growth rate is much smaller. And we have this first generation that survives much longer. Okay? So I'm not talking yet of those tendencies here. I will come back to them later. So in a few previous studies, John, the limitation of bacterial growth was explained by the balance between division, attachment, and erosion. So indeed, some the stronger the flow, the more detachment there is, and the less attachment. So what we did other experiments with high frame rate acquisition, so 10 Hertz. So much faster, okay? 1 minute to 10 Hertz, so 600 times faster. And we computed this ratio of detachment, number of detached particles over the total number, number attached over the total number. You see how it varies? So you have the statistics here, a median, a mean, plus minus sigma, first quartile, last quartile, and so on. So you see that the detachment increases strongly with shear stress, the attachment decreases strongly. But so what we see is that at U low, the bacteria will detach, but then they reattach nearly instantly. At low, 70%, 35% of the bacteria that detach will reattach. And then at high, less than 10% of the bacteria that detach reattach, and none coming from upstream attach. Okay, so we can actually, instead of looking at the cumulative rates, we can actually look at those logarithmic instantaneous growth rates for the detachment and the attachment and average them over time. And here you see how they vary. So there are those two things here. So you see that, of course, the detachment increases within the error bars, and the attachment actually is non-monotonic. And then we can correct this observed growth rate by those two things. John, that was the answer to your question. To get the effective growth rate, the one we would get if we didn't have detachment and attachment. And so what we see here is that, in fact, if we go from ULO to MED, we have 3/5 of this difference here in the growth rate that is due to the fact that there is less division, and only 2/5 that is due to erosion. But if we go from low to high, it becomes 3/4 due to less division, and only 1/4 due to erosion. But in any case, we can correct. Okay. So these tendencies here are actually the observed eta from the high acquisition frame rate. So they are from totally different experiments, and they are decently in agreement with this. Okay. So now, when we analyze this, the division of the bacteria, so we can analyze both the movement of the center of gravity here, basically its mean square displacement, and then we will analyze the division. So we want to detect the bacteria that never divide, and for this we actually follow this MSD, and if it's smaller than the half of the size of bacteria squared, we consider that it doesn't divide. Okay, because if it divides, the length has to double. All right. So we call that these bacteria a divider, and the other bacteria are non-divider. Okay, and so we're going to have a look at this heterogeneity in time. So the non-dividers are in yellow, the dividers in red. Okay, and those are actually this here should be eta f. So this is actually the corrected growth rate from the fast acquisition experiment, which is also consistent with those. Right. So what we see is that the fraction of dividers produces non-dividers. A fraction of dividers produces non-dividers because this population of non-dividers increases. They do not divide, so those which divide actually produce some that do not divide, right? But the fraction, if you divide this by this, or this by the total, the fraction of dividers decreases in time first, and it also decreases with the shear. So you see that at high shear, there is nearly no increase of the dividers. In fact, if you think of one divider at time t, so at time t plus its average division time, it will give rise to 2 bacteria. And among these, F bacteria, the fraction of dividers, so fraction f will be dividers. So that means that the number of dividers at t plus tau d is actually 2f times the number of dividers at t. So this is a geometric progression. So you get simply that the number of dividers at t is a geometric progression of 2f. That means the effective growth rate, the effective logarithmic growth rate that we have measured, that should be actually eta here and not mu, is simply ln of 2f divided by tau d. So you can relate this effective growth rate that we measure to the fraction of divider that we measure independently in the other experiments, okay, in the fast acquisition experiments. And that's, so we actually plot the fraction of divider versus the effective growth rate, and we get these things. So we have 4 different shear rates. So when we increase the shear rate, of course, we decrease the fraction of dividers, as we've seen, and we also decrease the effective growth rate, okay? And so if we plot this model, this very simple model here, it seems to be fitting, but I should say that we are using here a fitted parameter for the average tau d. And that fitted parameter is 52 minutes. So it's not so different from the 40 minutes I cited before, I mentioned before. And so given the uncertainties, it's not too bad. Okay, so that means that the decrease in effective growth rate with increasing shear comes from the decrease in the fraction of dividers in these systems. So now if we look at a single bacterium trajectory like this, you will have a divider like that, which its size will increase, because it's dividing. And then if you look at others, their size will remain constant. At some point they will increase. The size will increase, they will divide. So these ones, so the division time varies between 10 minutes and 300 minutes. Yeah. But the elongation rate during the division itself, the division event, is the same. Okay, so the division time that we measure is actually due to a lag time where they do not divide, and then the length of a division event that is the same for all of them. But the lag time differs. Okay, so we can then find among the dividers, we can distinguish the continuous dividers and lag dividers, and we see that actually the lag dividers, they're not very much impacted by the shear rate. So we see the evolution here, which is consistent with what we've seen before, of the fraction of continuous dividers and non-dividers. Okay. Okay, so now if we look at the statistics of this division time, that is lag time plus the time for the division event, we found those distributions at the different shear rates. And so at the earlier times, up to 160 minutes, what we have here is a Poisson distribution. So it's basically the distribution on independent events occurring at a constant rate, and with the constant rate being 1/40 minutes. So this is what we've plotted here. It's not a fit, but it seems consistent with the data. So that means that this white region here are the dividers, and those ones are the lag dividers. All right? And that distribution here for the dividers is not impacted by shear stress. So that's what we've seen and what I've mentioned before, that basically all division events occur in the same way for all bacteria. If we look at the mean division time for the different shear stresses as a function of experimental time, you see indeed that it decreases. It decreases because as time goes, as the bacteria divide, there is less and less lag dividers. So the lag time for lag dividers decreases. So between successive division, dividers have little by little get smaller lag times. Okay, so they have smaller lag times than their mothers, basically. So if we look at the two poles, if we consider the two poles of the bacteria, we are considering the attachment behavior. We know that they are attached by at least one pole and less strongly by another pole, potentially. So we look at this displacement here in micrometers of the two poles. So one in dark blue is the one that moves the most, and the other one is the better attached. Okay. So this is for continuous dividers for the fourth shear rates, lag dividers and non-dividers. Okay, so clearly we see that as we if we consider non-dividers, there is not much difference. So basically, the two poles are equally strongly attached, and that's actually why they don't divide. Okay. And the dividers have a pole that moves much more than the others. The lag dividers are kind of in between. And there is also, for non-dividers, so the non-dividers are attached symmetrically. And when we increase the shear, we get stronger and more symmetrical attachment. Which goes with the fact that there is more non-dividers. So what we demonstrate here is that basically the division and the attachment are two sides of the same story. So if the bacteria decide to use their energy to attach better, they will not divide or they will divide less. It's as simple as this. Okay, so if you want a little sketch to summarize, at the beginning we have three populations. We have continuous dividers in blue, lag dividers in green, non-dividers. So the dividers will divide and they will give rise to non-dividers, but as well as lag dividers. And at the end, you will have mostly continuous dividers because the lag time of the lag divider will decrease and non-dividers. And so the non-dividers are strongly attached at both poles. The continuous divider are weakly attached at one of the poles, and the lag divider are in between. Okay. So what we've shown here is basically that some bacteria choose to use energy to be more solidly attached, and this does divide less. So this response is non-homogeneous. So we have much more non-dividers for high shear rates than for very low shear rates. And those growth phenotypes are strongly correlated to attachment phenotypes, which is basically either they attach strongly or they divide. So there is here a force sensing mechanism that regulates cell attachment and growth rate. So the question is, what can we infer in terms of biological behavior? Because we basically use only physical measurements. So the phenotypic heterogeneity is known to be a key component of bacteria's ability to survive stresses, environmental stresses. So that could be, I don't know, antibiotics. In that case, it's shear stress. It arises from the individual response of cells to both the signal variability, so fluctuation in the signal, and also the stochastic fluctuation of the cellular machinery itself. Okay, so there are previous works who've shown that in the absence of flow, the effect of this stochasticity is seen in a heterogeneity of attachment. Okay, but what we show here is that in this case, the phenotypic heterogeneity is driven by the perception of a mechanical cue, which, to our knowledge, was not shown previously. And since the fraction of lag dividers with newly created cells decreases in time, it means that the dividers are able to transmit their phenotypic state to their offspring. So the offspring, when it is born, knows that it will have to wait less time before dividing. Okay? That's it. So what we postulate is that this type of growth arrest. So the growth arrest is something that is seen by biologists in what they call viable but non-culturable state of bacteria. So bacteria that are not dead, but they are not growing. So we think that this growth arrest and the enhanced surface attachment are two sides of the same response to stress, basically. Okay, and so another hypothesis that we can make is that this could be some type of bed hedging. And so bed hedging is the fact that a population of bacteria that are facing an uncertain future will lead them to develop different phenotypic behavior within the population, even though they are totally the same genetically. Okay, so this is a biological concept that apparently is trendy at the moment. So I would like to acknowledge, first of all, Antoine Hubert, because it was his PhD work, or part of his PhD work. Julien Faravin, who is the engineer who worked with the bacteria, who is a microbiologist. Tanguy Leborgne and Hervé Tabuteau, who is from another institute in physics, who supervised the PhD with me, and Alexander is a current PhD student of Tanguy who helped with the data treatment also significantly. And of course, we collaborated with a colleague who is an ecologist who's called Alexis Dufresne, and thanks to whom we could make some let's say. Most of this discussion basically was inspired by what he explained. Okay, so this was published in one. This is basically one publication that is in press at the moment. There is another one where that is a theoretical and numerical one, and that addresses the coupling to transport, diffusive transport. So. It's also interesting, but it's a bit more related to the initial aim of the PhD. I thank you for your attention. And just to finish, I'll just advertise two positions that we have in the autumn. There is a postdoc position on the characterization of hydrocarbon in situ bioremediation. Both by optical method in those millifluidic cells, the porous medium is here, and also coupled to spectral induced polarization, funded by the French National Research Agency. And there is also a PhD position on the subsurface storage of CO2 by dissolution. So both are experimental, mainly experimental. This one is funded by EUF and CNRS. So if you know anyone who could be interested, or if you're interested yourself, don't hesitate to contact me. Thank you.
Thank you, Yves. So is there any question?
So you found that this fraction of phenotypes changes with time, but this affects that simple calculation, no?
Sorry?
You have a simple calculation where you get the geometric progression.
Yeah.
So that won't be true anymore, right? Does it affect it much, or does it make a difference? So because you assume when you do that calculation, you assume that the mean division time is constant, no?
Yeah, so
That's not okay.
yeah, so actually it's all the truer as time goes, right? So
can you repeat? Maybe it's easier if I put. Maybe I'm just missing. So that was what I was talking about before, and then if you go back a bit more. Yeah,
so here we have a hypothesis that this division time, mean division time is constant. So we use the mean division time. We had a mean division time that was 40 from the data. It's just so it's very simple. Just we consider a mean division time. That's it.
Then if either f or tau d depend on time, this will give a different result.
Of course, then it's a bit more complicated.
I wonder if it gets you back to 40 minutes.
Maybe. So I mean, we could maybe put in some distribution that would be this this Poisson distribution plus some long tail, you know. And try to put this in and see if we can solve it analytically or at least numerically and get something that is a bit closer. But you see the data that we have, right? The data has huge errors.
I
So,
agree, it's good enough.
and this is actually not too bad, because it's so yeah. Yeah.
If there is no question, let's thank Yves again.